Hey guys, this is chapter 11, and so I'll go through the whole chapter just like I have before. And I have some fun backgrounds that I thought you would enjoy. I'm not actually on a plane right now, but I found a picture of an empty plane. I'll pop up another one halfway through. So this is the very last chapter that we're going to do for the year. And after this chapter, we'll work on our COVID-19 projects. But I tried to apply this as much as possible to what's going on in our world so you can have a better understanding of um, what the science is behind. So after this chapter, we will be working on our COVID-19 projects. So I wanted to start with kind of a connection from chapter 10 and something that happened to us in our lives. So here's an example that happened to us, and I'm sorry this is choppy. I've been interrupted a bunch of times by my kids, so I don't know where I am. So we are hatching chicks right now. We hatched like 20, and we've got another round coming, and I just ordered a bunch, so we're just doing a lot of chick stuff. But we had something happen that's never happened before. So here's a picture of it, and you can see this little baby, and on the picture on the left has a couple webbed toes. And I've never seen this before. It's only between the two outside toes. Chicks have 4 toes, 1 in back and 3 in the front, and so it's just between those two. And the little guy was having a lot of trouble walking, and he also had like a weird eye problem where his eye like lid wasn't big enough, and so his eyes didn't quite open all the way, and he had a hard time shutting his eyes all the way. So we could tell that something was wrong, that he had some kind of a mutation. And actually what happened was his body didn't do apoptosis. Remember what that word means, where we have programmed cell death. So like when we're babies, we start out with webbed hands, and then we do apoptosis and we kill all these cells between our fingers. And so then we're able to have fingers. And that happens other places in our bodies too. Apparently it happens around our eyelids. So he had a problem with apoptosis. And we ended up cutting that. Well, Jay did, and it was awful. He slit through that little web, and he was able to walk a little bit better, but he still wasn't well. And so this is a picture here at the bottom of the next day, and he ended up dying that next day. So what I think happened is that there was probably other things wrong with him inside. Maybe some other issues, maybe apoptosis didn't happen. But I wanted to show you that because this is an example of a mutation that was not beneficial. This little guy couldn't walk, and maybe couldn't see well, couldn't close its eyes, maybe had other things happen. So this mutation was not beneficial. And it wasn't that the environment caused it. He was just born with a random mutation. And we wouldn't really want other baby chicks to have that. So we maybe wouldn't want him to even have babies because he wasn't able to live. He kind of wasn't compatible with life. And so this happens sometimes in living things, but sometimes we also have mutations that are good, that help us to live better. So both things can happen. So in section 1 here, we're looking at genetic variation and how that can change the chance of individuals to survive. And so that's kind of what we saw with this little baby chick. We called him Webster because he had webbed toes. So we find that if there is a genetic variation, then their phenotypes might look different. In this case with Webster, he had a different phenotype. He had webbed toes. And we need phenotypic variation. We need for them to look different and be different in order for natural selection to act on them, just like they did on Webster, and he died. And this genetic variation is in the gene pool. And so we look at the alleles to determine what different types of genes are in the gene pool. Is there a gene for webbed toes? And so we can actually count the alleles in a population sometimes. Oops, I thought I had this on here. So here we have some green frogs and some brown frogs. And we could actually, if we assume that the brown frog is recessive, we could actually count how many alleles are in the population. This is similar to what we did for the blue ones case study, where we were actually like writing down their genotypes and predicting what genotypes they had. My kids also wanted me to show you how Twilight Sparkle. This is Rye's hamster, and she is biting me right now, but she has diabetes, we think, because she pees all the time and drinks tons of water. And she also has a big eye. It's kind of swollen, and so we think that might be glaucoma, which is associated with diabetes in hamsters. And so genetically, these little dwarf hamsters get diabetes a lot. So we kind of thought that was interesting. And hopefully she has a good life and can just live her life. And we try to keep plenty of water and change her little cage. So that's Twilight Sparkle with diabetes. Okay. So let me, I gotta go wash my hands. Okay, so if we continue on here, we can look at these allele frequencies and look at genetic variation. So if we have like an even spread of allele frequencies, then we're going to have more genetic variation. So we're looking at how common is an allele in a population, how common is this webbed foot trait, how common is the diabetes in hamsters, and we can calculate that for the gene pool. So here's the picture I was just thinking of. If the brown frogs are recessive and we've got 2 of them out of 6, then we can actually calculate how many alleles there are. And out of the green frogs, some of them are going to be big G, big G. Some are little g, big G, heterozygous. We don't know unless we do a test cross, but we can predict kind of that would be half and half, or a 1 to 3 to 1 ratio, or something like that. And then we can try to predict how many alleles are in the population. So that's one way that we look at genetic variation. And we can get this variation from random mutations like Webster, and they can form a whole new allele sometimes if there's a mutation, and that can sometimes be passed on to the offspring if it's in their sperm or egg. And we can also get it from recombination, from crossing over other mutations that we learned about in chapter 8. Usually that would be in meiosis. Okay. And so also we can have hybridization of 2 different species. So 2 individuals that wouldn't normally be able to mate but are really close species, they can actually mate together and sometimes form some variation. So now we're going to go very quickly through some of these questions, and if you want to skip this, you can. So the first question is, a measure of how commonly a particular allele occurs in the population is known as, and that is allele frequency. And then the more genetic variation a population has, the more likely that some individuals will, so it'd be survive. We want variation in our chicks, so we have lots of traits. Genetic variation can arise from a random change in the DNA of a gene, and this is called, what's that random change called that can happen? That is a mutation, which term means the crossing of 2 different species that share common genes that would normally mate. And so that would be hybridization. So now section 2 is looking at populations and how they can change. So remember, when evolution happens in natural selection, it's not happening to one individual. It's not really even happening to Webster. It's happening to a whole population, many organisms, all of these penguins, and over many generations. So we're not just looking at an individual. So this is what we normally see when we look at populations. We normally see that most of them are kind of in this middle medium range here, and few of them are on the extreme. So for example, most of us are average height, and then there's a few people who are super tall and a few people who are super short. With our chicks, most of them have normal feet, and then maybe a few of them on one side would have webbed toes, and then maybe on the other side, they do have some webbing like us. So maybe on the other side, they don't have any webbing. Maybe there's too much apoptosis. And so usually we see this, what we call a bell-shaped curve, and this is a normal distribution. And like it says here, the mean value is in the middle, and we have the most, and then we have fewer on the extremes. So there's 3 different ways that natural selection and populations can change. But one way that we look at is microevolution, and that's where they just have like small changes in allele frequencies, just like Webster, that aren't changing, like they're not changing into a different species. They're not changing drastically. But they can also have one of these 3 directions, or types, I guess I should say. The first type is directional selection, and this is where, so the dotted line is where we're starting, and then the solid line is where it's shifting to, and this is where we see a total shift in the traits. So the average trait becomes kind of the median, or one of the extremes. And so we see this with antibiotic resistance in bacteria, if we start taking antibiotics, then we're going to shift it to having a high population of resistant bacteria. So they're shifting towards the extreme. So that's directional selection. Stabilizing selection is where they are shifting more towards the middle. And this is kind of a cool example that's in your book. It's talking about gall flies, I think. Yeah, and so these birds, these woodpeckers, they prey on the large galls, and the wasps, I think, lay their eggs. These are like gall flies. They lay their eggs on the small ones, and so they're kind of the big ones and the little ones are getting affected more, and so the medium ones are the ones that are gonna produce more. So they're gonna have a higher mean. So this is stabilizing selection. And then the last is disruptive. And so in this one, you're favoring the extremes and not the mean. So this is a good example here where you have buntings, which are these little birds, and the little ones, so the big ones are able, no, this is blue and brown. The ones that are very blue are able to mate, the males. But then there are some males that are kind of in the middle, bluish brown, and then there are some that are very brown and plain. And the blue ones that are competing for the mates, they're going to ignore the brown, boring ones because they don't think they're going to get any mates. But in fact, those are the ones that are sneaking in and mating with the females. And so the blue ones are competing with the medium ones, but they're ignoring the plain ones, and they're actually getting mates. So that's kind of a fun directional selection idea. So let's go through these questions. What is the observed change in the allele frequencies of a population over time called? And so when we look at just a small change in allele frequencies, that would be microevolution. Natural selection that changes the distribution of a trait to favor one extreme phenotype is called, so we're looking at an extreme one, it's shifting to an extreme, that is directional selection. In stabilizing selection, what occurs in a population? So think about that graph with stabilizing selection, that second one. And that would be the intermediate phenotype becomes more common. The next one here, a population that is not undergoing natural selection displays what type of distribution? So if it's not having any kind of natural selection, that would be normal distribution. And I drew a picture for this one. In a population of birds, intermediate beak size is selected against, and both very large and very small beaks are favored. What type of selection is this? And so I drew, this was a tardy bell thing, first period tardy bell. Most people come in between the first bell and the tardy bell. And so that would be disruptive selection. So section 3 here is on natural selection and how it actually operates. And there's different things that can happen for natural selection to happen. And some of these terms are kind of confusing, so let's try to keep them straight. One way is through gene flow, and that's when genes are moving between populations. And this can happen through migration. And here's an example. We're looking at these hatching areas of bald eagles, and they have like different sort of populations. But if they're able to mix and mate, then we have gene flow. We have migration and gene flow. If they can't mix and mate, we don't have gene flow, and that's a problem. We do need gene flow. We need gene flow in human populations too, which is why migrations are good in human populations and people coming in from other countries to kind of keep that gene flow moving. Okay, another term is genetic drift, and a lot of times people get gene flow and genetic drift mixed up. Genetic drift is just random. So think of drift as random. Gene flow is migrations, genetic drift is random. Genetic drift can cause a loss of genetic diversity. It happens most in small populations, and this is where some of them just die. You can get like a bottleneck, and some of them just die off. It's not any particular reason. It's not that one trait is better than another. It's not like that. It's just some of them die. And this is a picture of what that might look like. So bottleneck is a good example. Bottleneck is when a whole bunch of them die off for one reason or another. Maybe it'll happen to the human population here with COVID-19. Hopefully not. And it's not that there's a particular trait that maybe makes one better than the other with this example. And then this is going to limit your alleles that are left. So in this example, you don't have any red alleles left. Founding effect or founder's effect is one way that genetic drift can happen. So this is where like you start a new population from another one, and it just randomly, whichever traits make it up over to the new population, but you're going to get a decrease in genetic diversity from this. So definitely negative effects on the population because you have less genetic diversity from genetic drift. So remember, this is just random. Doesn't mean that any traits are any better than another. Sexual selection is another mechanism that can cause natural selection, and that's when there's certain traits that make an individual more likely to mate than others. So most of the time it's the female choosing the mate, but sometimes that's not true. The female has a higher cost, so the female puts a lot of energy into making an egg, and the male puts it makes a lot of sperm, so maybe it's equal energy, but there's just more invested in the female's egg, so she might be more choosy for that reason. And she also might be more limited in when she can reproduce. Some animals can only reproduce like certain months of the year, like birds. And so this is a male bird here, and you can tell he has really showy tail feathers, and so that might be something that the female would prefer in the male. So that would be sexual selection. They're choosing something. And there are two types. There's intrasexual selection, where the males are competing, and so we're talking about the same sex is competing there. And then intersexual is when the males are displaying traits like that showy tail or like this lovely throat. So those are some mechanisms of how natural selection can occur. Now let's go through the questions. I'll go through them quickly. Chance changes in allele frequencies within a population are called, so what are those random chance changes? Those were genetic drift. And then a small number of birds blown off course during migration find an island and colonize. This population will most likely experience genetic drift as a result of gene flow. So that's migration, and that is going to cause genetic drift. It's a founder effect there. Fighting between male elephant seals over females is an example of what? So that would be intrasexual selection because it's within the same sex. What increases genetic variation when animals move from one population to another? So it's migrations. What's going to increase genetic variation? And that would be gene flow. Okay. And a problem in all populations influenced by the bottleneck is that, so what's the problem with bottleneck? We are going to lose genetic variation for sure. So that's the last question there, and then we'll move on to section 4. All right, here's the next one. 11.4 is on Hardy-Weinberg equilibrium. So these are two people that were working on looking at how populations change and how allele frequencies change. They came up with this model that we'll look at that's pretty helpful, and it ties in with the last section really well. So we're really not learning anything new, we're just kind of applying it. So basically, Hardy-Weinberg equilibrium is like a set of rules, and if all of these rules are true, then the population is not changing. And basically, there is probably not a population on Earth that exists that's not changing. So there is no population that really follows these rules. Here are the rules. So if these 5 conditions are met, the allele frequencies are going to stay the same. So first one is if there's a very large population. If there's a large population, that means there's no genetic drift. The second rule is there's no emigration or immigration. So that would mean there's no gene flow because that's from migrations. The third one is no mutations. So if you're not adding any new alleles to the gene pool, you're not going to see any change from genetic mutations. The fourth one is random mating. If there's no sexual selection and there are no traits that make some organisms more or less likely to mate, then that would be random mating. And then the last one is no natural selection. So all the traits are equal. You don't have any webbed-toed chicks. All the traits have just an equal chance of surviving. If all 5 of those things are true, then the population will stay in equilibrium and it will not change. But this never happens. So I want you to think about, like, well, what's a situation where one of these is true? Like, the human population is very large. We do have probably very little genetic drift. And but we have immigration and emigration, and we definitely have mutations, and we definitely don't have random mating. There's some traits that make mating more likely than others, and we probably do have natural selection in humans as well. You can think about it with these penguins too. Okay, so real populations, that rarely meet all 5 conditions for very long, if at all. And also the environment's always changing. So that's going to cause natural selection, that 5th one, to happen. So we can use this to predict some genotype frequencies. And I don't think we'll spend a lot of time on this, but we actually use Punnett squares. And Hardy and Weinberg came up with a pretty cool equation, this equation here, to look at populations. And if you take AP Bio, we'll look more into this, study that more. Okay, so here is just a review of the last section. Genetic drift is just random changes, and look, we wiped out the whites and the greens. And then gene flow is we have some coming in and leaving, so that would be immigration and emigration. Then we can have mutations. Oh, look, we have a new allele, so that would be mutations. And also sexual selection. So some of the traits, maybe a blue body color, make them more likely to mate than others. And then the last is natural selection. So some traits make them more or less likely to survive. And you can see that change in allele frequencies there. So we know that in nature, populations do evolve. They change in response to the environment. They're changing all the time, and populations never obey those 5 rules. So they're always changing. So let's go through these questions. Which of the following conditions could mean that a population is in Hardy-Weinberg equilibrium? So which of these would be true for them to be in equilibrium? We would have random mating for it to be in equilibrium and not change. Hardy-Weinberg equation is used to do what? So there's our answer. We're predicting genotype frequencies in a population. The 5 factors that can lead to change are gene flow, genetic drift, mutation, natural selection, and sexual selection is the last one. And then mutations are important because they bring about, they bring new genetic variation to the population. Sometimes it's good, and sometimes, like Webster, it's bad. Which of the following statements applies to real populations? It is rare for a population to be in Hardy-Weinberg equilibrium. All right, we'll go on to section 5. All right, so section 5 here is looking at new species and how they can arise, and this is called speciation. So we'll look at different methods of speciation here. One thing that can happen is that if populations become isolated when there's no gene flow, so when they're not in Hardy-Weinberg equilibrium, they can actually change and become different from the other population that they were isolated from. And so they're going to adapt to their own environment. So imagine, like, if you had an island that broke into 2 and you had a population of birds, if they weren't able to mate anymore between the 2 islands, they could eventually change. And these are some of the things that Darwin saw, like with the tortoises on the Galapagos Islands, and he saw that those were different eventually, and they couldn't mate anymore. We can also have reproductive isolation occurring, and that's when different members of the population cannot mate anymore for different reasons. So here's an example here. These are fruit flies, and if these fruit flies start to maybe eat different things or be in, like, different, here we have different climates, eventually they won't be able to mate anymore. And we can get new species arising from that. We can also have behavioral barriers. So maybe they have like different courtship dances, or they start to have different mating behaviors like scorpion dances or other bird calls, other behaviors. So eventually, if they don't have those same behaviors, they're not able to mate with each other. So that would be a behavioral barrier. And then a geographic barrier, that's called geographic isolation. That's like the island I gave as an example. If an island split up, or if you have like the Isthmus of Panama, like if you have a barrier that is preventing them from mating. Another example is there's two like squirrels in the Grand Canyon, and they're separated by the Grand Canyon, and so they're very similar, but they live on opposite sides of the canyon and they can't meet. So that would be a geographic barrier or geographic isolation. And then temporal barriers is another one. If they mate at different times of the year or different times of the day, maybe one is nocturnal and the other one's not, that would be temporal isolation, and they can't mate, and they might eventually become different species. So let's go through some of these questions. So the first question is, what is speciation? And remember that we're looking at, there's a typo there, divergence of two or more species from one. And this is what Darwin first saw. This was his first clue that something here is going on. The second one, a difference of chemical sense between two populations, is an example of what kind of isolation? So we're looking at behavioral isolation, and that can cause like a sexual isolation where they can't mate anymore. An earthquake causes an ocean channel to open up on an island where a low area previously existed. The lizard's population is now separated on two different parts of the island. What type of isolation is this? So this one would be geographic isolation. Which of the following examples illustrates a temporal barrier between mating populations? So we're looking for temporal, which is time. Which one would be a temporal barrier? This one's a really good example. So take a minute and read through those. So varieties of oak tree produce pollen during different seasons, and they can't pollinate. Even if it's a difference of a few weeks, it might matter. What kind of isolation occurs when two populations of birds have different courtship dances? So if they're dancing differently or have different calls, what would those be? That would be a behavioral isolation. So now we're finally on to section 6. So in this section, we're looking at different patterns of evolution. Does it happen fast? Does it happen slow? Remember, evolution happens through natural selection, through genetic drift, through gene flow, through sexual selection. All of those mechanisms that we looked at. It's not just natural selection. That's just one of them. And it's not random. It can go in a certain direction, and these effects can add up over time. And we see that with COVID-19, with SARS-CoV-2. It also went through natural selection and eventually became a really effective virus. So here is an example of that. As these effects can change over many generations, we can definitely see changes in the population, and we can eventually see speciation, where it turns into a different species. There are different types of evolution too. Convergent evolution is where different species evolve together. So you can see this moth and this flower. They have a mutualistic symbiotic relationship where the moth is going to get food from the flower and the flower is going to get fertilized. And you can see that they evolved together to be very specific, to kind of fit each other. So that's an example of convergent evolution where they change together. Divergent evolution would be where you have one organism, like an ancestor, that would diverge into two different organisms. And so that would be like if we saw a temporal barrier or a geographic barrier, and then that one ancestor diverged into two, and they became different. Those are different types. We can also have coevolution. That was that picture I showed you. And in this case, they're evolving together. So that would be the moth and the flower. They're responding together and they're evolving together, which is really interesting. And there is an example in your book with this ant that evolved along with this little plant, with this type of shrub, and they have a beneficial mutualistic relationship that's symbiotic. They're helping each other. And then sometimes coevolution can happen in competitive relationships. So here we have this crab that is preying on this snail. And so over time, the snail gets kind of pricklier and spinier, and then in response, the crab is going to get bigger claws to try to respond to that and still be able to prey on the snail. So sometimes it can be competitive, and sometimes it can be beneficial. Extinction also happens, and that's kind of part of evolution. That's when a species is completely eliminated from Earth, and we've had a lot of really big extinctions, like really big extinction events, but we've also had background extinctions which happen at a low rate, kind of in the background. Every once in a while we'll have something go extinct. And that should occur at the same rate as speciation. So as we're losing species, we should be getting new species. But recently we've been losing and having more extinct species than normal. Okay. Yeah, so we want them to occur at a slow rate, usually in small areas, but sometimes they happen more frequently. And it is possible that right now we're going through a mass extinction where we're seeing a higher number of extinctions than we have in the past. So mass extinctions are rare, but they have happened, and that's where we lose like a really large proportion of the organisms on Earth. That would be at a global level. And here are some different mass extinctions that have happened. So the peaks are the extinction rates in the last few 100 million years. Okay, and then we also look at patterns of speciation. Sometimes it's punctuated, which means that it happens like in short jerks, like boom, lots of speciation, and then it dies off, and then lots of speciation. This was a theory from Eldridge and Gould in 1972. And so sometimes we see those big events, but sometimes it's slower and it's not as punctuated. So we might see both. And here is an example of what we look at when we see ancestors that are branching out to their descendant species that they're related to, and that's because of adaptations to their environment. So we can really see them adapt and form different traits. All right, let's go through the questions. Which of the following processes is considered to be random? So which one would be random? Genetic mutations would definitely be random. They just randomly happen. They might be good, they might be bad. The American flying squirrel and the flying phalanger of Australia live in similar environments and look very similar. However, they are not closely related. Their resemblance is most likely an example of what? So they don't look the same, or they look the same, but they're not related. That would be convergent evolution. The Galapagos Islands in the Pacific Ocean have varied habitats. Divergent evolution is occurring on these islands, meaning that the species that live there. So this is what Darwin saw with the tortoises and the finches. And he found that they had a common ancestor. And then as they diverged, they became very different and couldn't mate together anymore. Like all flowering plants, the snapdragon must be pollinated to reproduce, but its flowers are closed. A bumblebee has just enough weight to open a snapdragon flower by landing on it. This adaptation is an example of what? So this would be coevolution, where they're evolving together. The total and permanent disappearance of a species from Earth is called, that is extinction. Oops. Did I go too far? Oh, that was the last question. All right, there we go.